back to Second International Brain Environment Conference, IBBC 2024. And I'm Larry Nawaz. I will be the moderator of this session. In this session, we will talk about biotechnology, microbiology, and physiology. So for our first oral presentation, I would like to call Dr. Mavish from GCUF, and she will be presenting on bioprotection of maize by plant growth promoting bacteria against Aspergillus flavus.
Bismillahirrahmanirrahim. First of all, I would like to thank the organizers of this conference. That is the Second International Brain and Biomedicines Conference organized by Department of Physiology, Government College University, Faisalabad. I'm Dr. Mavis Salman, working as Assistant Professor in the Department of Biochemistry, GCUF. The topic of my presentation is the bioprotection of Z maize L by plant growth promoting bacteria against Aspergillus flavus. These are the contents of my presentation. Let's have a brief introduction of my presentation. The plant growth promoting bacteria. These are termed as PGPB or plant growth bacterial species that enhance plant growth and also protect plants from biotic and abiotic stress. These plant growth promoting bacteria also decrease negative environmental effects of chemical fertilizers, pesticides, and microorganisms, and also act as biofertilizer. Pathogenic microorganisms, including various genera of fungus, bacteria, viruses that have adverse effect on maize growth, and they can also cause inhibition of plant seed germination. And they also cause a disturbance in physiological and biochemical parameters, reduce yield, and also produce mycotoxins. This is a flow sheet of methodology performed in this project. First of all, we have grown the bacterial species, the bacteria which we used in this study as as a treatment is the Cucaria oceani, which is isolated from rhizosporic soil. First of all, we have identified the antimicrobial potential of that Cucaria oceani species. The sequence of that species has already been submitted in NCBI Gene Bank. And then we have collected maize cultivar, that is Melcar 2016, from Ayub Agricultural Research to conduct the pot experiment by bioprime our maize seeds with Cucaria oceani against Aspergillus flavus. After bioprotection experiment of V maize L, we find out the various growth parameters in parallel with various biochemical parameters, including phenolic flavonoid contents, proteins and sugar contents, and antioxidant enzyme measurements. And whereas the growth parameters include root and shoot length and the biomass of root and shoot. These are the different treatments with codes that are applied on maize seedlings. These codes termed as T0, that is the unprimed treatment, means that maize cultivars are not treated with our selected bacteria, that is Cucaria oceani, and even not infected with fungus. These are termed as the controlled seedlings. The second one is T0 bar. It is the treatment named as unprimed seeds of maize infected with Aspergillus flavus. And the third word treatment is T1. That is the maize seeds bioprimed with Cucaria oceani plus infected with Aspergillus flavus. For each treatment, there are 3 replicates. 3 seeds were sown 3 centimeter deep, and after 14 days, samples were collected from pots, and the experiment was performed in completely randomized design. Now we move to the results of our experiment. This is the antimicrobial potential of bacterial strain. Our potential bacteria is Puccaria oceani. As I told earlier, it is extracted and isolated from rhizospheric soil of maize roots. The culture supernatant of Puccaria oceani performed the inhibition zone in millimeter against the pathogenic strains. These are given in including various bacterial and one of the fungal pathogenic strain, that is the flavus, which is the main infectant in our study. This figure represents the pot, that is the T naught. This one is the center one pot is the T naught, that is the unprimed seedlings plus Aspergillus. These are the unprimed seedlings, whereas the T naught bar is the unprimed seedlings plus infected with Aspergillus flavus, and this T1 is the bioprimed seeds infected with Aspergillus flavus, and you can visualize the effect of bioprimed seeds, even in the presence of Aspergillus flavus. These are the different growth parameters: root fresh weight and shoot fresh weight and shoot dry weight. You can visualize distinguishing characteristics between the T naught bar, and T1. Similarly, these are also the growth parameters: the root dry weight, shoot dry weight, and the root length and shoot length. And you can observe that the T1, that is our primed seeds in infected plant. These are the chlorophyll contents: chlorophyll A, chlorophyll B, and total chlorophyll contents. And the same pattern has been observed in these graphs. That is, the minimum chlorophyll contents were found in T naught bar, that is or unprimed seeds infected with Aspergillus flavus, whereas the T naught are the unprimed seeds. These are the control, and the T1 are our treated seeds with Puccaria oceani. These are the MDA and hydrogen peroxide contents, and you can visualize that the MDA and the H2O2 contents, these are maximally observed in T naught bar, whereas the total soluble protein and phenolic contents were found maximum in T1 treatment. Similarly, the antioxidant enzymes have significant increase of the enzymatic activity of SOD, POD, and CAT in T1 treatment. After performing these biological parameters, we have also detected the aflatoxins present in our different treatments: the unprimed control, the unprimed infected with Aspergillus flavus, and primed with Puccaria oceani and infected with Aspergillus flavus. You can visualize that the AFB1, AFB2, AFG1, and AFG2, no concentration in nanograms per gram was detected in unprimed control, whereas unprimed infected has concentration of these aflatoxins or 4 aflatoxins, whereas the primed seeds with Puccaria oceani and aflatoxins, there no detection of AFB1 and B2 was found. However, AFG1 and a minute quantity or concentration of AFG1 and AFG2 was found. Let's conclude the overall experiment. The current investigations highlighted the use of multifunctional Puccaria oceanae to protect maize plant infected with Aspergillus flavus by improving photosynthetic pigment, increasing the accumulation of phospholites and antioxidant system of plant. Furthermore, biostimulants reduce oxidative stress in maize plants, reflected by the production of superoxide, hydrogen peroxide, and MDA activity. This work demonstrated synergistic bacterial interactions to explore affordable, eco-friendly biofertilizer for farmers in future. Therefore, field trials are suggested for further studies to understanding the potential use of plant beneficial bacteria for sustainable agriculture in fungal infected lands. Thank you so much, and be kind to your mind.
Thank you so much, Dr. Mavish. So we have next presenter with us, Dr. Kuratulain Gilani from WM, and she will be presenting on anti-obesity potential and toxicological evaluation of Pepervum somniferum seeds in obese rat model.
Dr. Kuratulain Gilani, and I am an assistant professor in Women University. The presentation which I'm going to deliver today is anti-obesity potential toxicological evaluation of peppermint somniferum seeds in obese rat models. As is generally known that obesity is itself a disease, and we define the obesity as a person whose body fat percentage is dangerously high and does not reach the BMI less than 20. Obesity can cause so many other diseases like heart diseases, increase the risk of heart diseases, blood pressure, uh, diabetes, cancers, and it's also very dangerous for your health, mobility. It's reduce the mobility of the human beings. Obesity can cause by three different effects. One is the genetic effects. We have some genes which can control our obesity, and due to which obesity can cause in families, and some have the abnormal hormonal responses. These hormones cause problems to body physiology and increase the body weight and cause obesity. And third form is the obesity is due to the high consumption of the junk food or high fat diet intake. A high fat diet cause so many problems. It clogs your arteries. It can increase the your heart problems, increase the blood pressure. In other terms, if obesity can cause all these problems. Now, the medicines which are available in the market are normally very high cost, and they have a very dangerous health problems or side effects. There are many plants or herbs which are used medicinally to reduce the obesity, and journals generally in many medical pharmacological companies use these plants. The one plant which I'm talking about is the Pepervum poppy seed plant or poppy plant, Pepervum somniferum. This plant is used for the collection of the opium, and the seeds of these plants called the poppy seeds or the hash. Hash is very common in the households, used in many recipes. It also has a very alpha linoleic acid in it, which can help in the reducing the obesity. It's omega-3 fatty acid, and it can be helpful to reduce the obesity. In our research, the main objective was our research was to see the efficacy of the papaver somniferous against the obesity, and check the toxicological effect on the liver or kidney. If uh, it is useful, it is safe to use as a obesity for the obesity. So in the first trial, we induced the obesity in the animals. So our first part of the experiment, in which we divide the group, one is the control, which have the normal diet, and the second with the high fat diet, in which we feed the animal with the breads, with the high fat and animal fats, and all those protein fats which can cause the obesity to the animal. And the feed was prepared with the percentages of these fats, and then we give this feed to the animal for the 6 weeks and induce the obesity. Then we divide these groups, this fat group, into further 4 groups, and then one became the control positive, in which have obesity, but we didn't give any type of treatment to the animal. In standard group, we give the animal the medicine which are available commercially in the market. And the third and fourth group was treated with the poppy seeds, but in this 2 groups difference was that one is washed poppy seeds and other was unwashed, because poppy seeds has opimium on it, and when we washed it, it's wash away the opimium layer on the poppy seeds. So after the dose was which we give is the 30 gram of feeds, 0.5 gram per 30 gram of the feed we gave on the daily basis to the animals. After that, we dissect the animal and collect the blood, and we did the experiments like we did lipid profiling, blood glucose test, which we did throughout the experiment, and the liver function test, kidney function test, CBCs, and also we did the histology of the animal with the kidney, liver, and the obese. Then we did the statistical analysis, 2 extra studies which we performed side by side of this experiment. We use this effect, the poppy seeds on the saliva digestion. Saliva has the enzyme called amylase, which digests the carbohydrates. So we use this amylase, and we see that if poppy seeds reduce the digestion, or did not have an effect on the digestion of the carbohydrates. And second was the behavior study. In first group, the result, the obesity induction trial, in which we divide the group 2 into and give a normal diet to this control group, and high fat diet to this group. And as you can see in this graph, that weight is definitely increasing due to the obesity, weight gain, and it is statistically very significant result. In second group, we divide these animals and into 5 groups, and then we give poppy seeds, standard medicines to 3 groups, and the 2 was our control negative and control positive for 4 weeks. It shows that unwashed poppy seeds reduce a little bit weight in week 4. So there is no very highly significant result in other groups, but it still have some effect on the weight, very low effect on the weight reduction. If we see that the effect of the poppy seed on the glucose level during the experiment, we monitor the glucose very carefully. As you can see in this graph, the value is highly statistically significant. and that the washed and unwashed animals have very low glucose level as compared to other groups. Again, with the HDL levels, which is high density lipoproteins, these three groups have low levels as compared to these two control groups. Again, in the low density lipoproteins, again the result was same and significant. That it's reducing the low density lipoproteins, and also it's reducing the very low density lipoproteins, and this value is very highly significant. Uh, Again, there is no very prominent effect of poppy seeds on the liver functioning or kidney functioning. Overall liver function or kidney function of the animal show the same results, and the damage of the kidney or liver is due to the obesity which we caused by giving the animal high fat diet. So there is no other effect on the animal on of which are given with the poppy seeds. The other experiment which we did on the side wave, in which we study the washed and unwashed poppy seeds effects on the digestion of the carbohydrates. And in this experiment, you can see this A test tube have a glucose and iodine. In B, have a glucose solution with amylase, and in C, we have amylase glucose solution and poppy seed solution. So you can see it's slowing down the digestion. It's not completely digested. This picture is taken after 15 minutes. This picture is taken after 30 minutes, then 45 minutes, then 1 hour, and then 1 day. And all these pictures show that it's again reducing the time of digestion of the carbohydrate by amylase. In this pictures, these are the washed poppy seeds, and again these shows a slow digestion of the glucose by the amylase. That's mean poppy seed and in the washed form are more effective for the carbohydrate digestion inhibition. So it's inhibiting the digestion of carbohydrates, which is a very common factor in the high fat diet, and it's increasing weight because carbohydrates are more generally used as energy production and production for the energy production. So it accumulate more easily. The other group test in which we did the rota-rot test to see the mobility of the animal, and it's very obvious result that control group which is not obese have a very high very effective on rota-rot, very mobile, and obese and treated group is not very mobile in this experiment. And in the open field also the mobility rate is low in the obese animals, but it is high in the control group and the group which is treated with the standard medicines for the obesity. The other experiment which was the comparison of the noble object recognition, the familiar objects, the results show not very significant effect on any of the groups, and with the unfamiliar objects there is not very statistical difference between the other objects also. So it means there is no particular effect on the learning behavior of the animal. So in the conclusion of this experiment, we can say that poppy seed help in reducing blood glucose, HDL, LDL, VLDL, and obese rats, which are on high fat diets. Poppy seeds did not exhibit any adverse effects on the health of the obese rats, and poppy seeds slow down the digestion of the carbohydrate by amylase enzymes. So there is no adverse effects of the experiment on the learning behavior on the animal. So it is very useful that we can use these poppy seeds for the obesity reduction because this the percentage we use, the concentration we use, amount 0.5 gram is very small. So in future we can use a high dose of this poppy seeds to see the effect on the obesity, or we can produce extracts of the poppy seeds and we can use oil of the poppy seeds, and we can see what kind of effects they can cause on the obesity. Thank you very much for your time. Now,
Thank you so much, Kuratulain. And our next presenter is Jumahan from JUAS, and the title of presentation is Exploring the Immune Potentiating Role of Iron Oxide Nanoparticles Supplementation in Broilers.
Bismillahirrahmanirrahim. My name is Juma Khan. I have done my post-graduation in physiology from University of Veterinary and Animal Sciences, Lahore. During my post-graduation study, my research topic was exploring the immune potentiating role of iron oxide nanoparticle in broiler. The study was conducted in Animal Shed of Physiology Department. As we know that the population is increasing day by day, so then their need of protein requirement is also increasing. The poultry meat is important source of protein to fulfill the demand of the increasing population. For the proper growth and development of poultry birds, feed additives play important role. As we know that poultry birds are fast grower, so they need proper feed additives in their feed to fulfill their requirements. Deficiency of feed additives predispose these fast-growing birds to various diseases which negatively affect their growth as well as production. Among these feed additives, iron plays important role in development as well as proliferation of B and T cells, which are important for the humoral as well as cell-mediated immunity respectively. Iron Iron nanoparticles have unique properties due to their small size. They have more bioavailability and also other properties which are used in other biomedical studies. Hypothesis of our study was that the iron nanoparticles supplementation improves the histological features of the sickle transit, which play important role in production and as well as immunity development of the T cells. The objective of the study was to study the relative organ weight of sickle tonsil and Barsat and Vicus lymphocyte proliferation and histology of the sickle tonsil. Nanoparticles were prepared in the lab of physiology department by using ferrous chlorate and iron sulfate at the molar concentration of 0.25. And then they were combined, ferrous chlorate and iron sulfate were mixed and in the presence of nitrogen, and then sodium hydroxide at concentration of 6 molar was added, and then they were stirred on the hot magnetic plate stirred for 40 minutes. And then finally they were centrifuged at 6000 rpm for 8 minutes, and finally they were dried by keeping in oven at 60 centigrade for the 24 hours. The characterization of the nanoparticle was done in UAT Lahore. Our study design was a bird, 300 birds were utilized and they were divided into 6 groups. Group 1 was taken as a negative control and were given no iron, and group 2 was given iron in bulk form, and the rest 4 groups were given iron nanoparticles. Group 2 was given 80 mg per kg iron in bulk form. The it was given according to the NRC requirement that poultry bird iron requirement is 80 mg per kg of feed. And the purpose of the subgrouping of the nanoparticle was that to observe the comparable effect of each concentration with the bulk form, so that we can determine that at which concentration of nanoparticle we obtain the optimum effect as we observed in nascent the bulk group, so that we can minimize the iron losses as well as the expenditure. The trial was carried out for 35 days, and at the 35 days, 2 birds from each replicate. Each group was further divided into 5 replicates, and each replicate was contained 10 birds. And at the end of trial, at the 35 days, we took 2 birds from each group and then slaughtered, and the sample was obtained. Parameter studied was relative visual organ weight, bursite, sickle tonsil, lymphocyte proliferation, and histology of the sickle tonsil. In histology, we studied number, length, width, and area of sickle nodules. Statistical analysis was done by using one-way ANOVA. Due to supplementation of iron nanoparticles, the relative bursal weight was observed while increasing the concentration of nanoparticles, and maximum effect was observed in group 3 and 4, which were given 20 mg and 40 mg iron nanoparticle respectively. At higher concentration, the relative weight was decreased, which shows that further increase in concentration of nanoparticles has no pronounced effect. The nanoparticles effect on the relative weight of sickle tonsil was against the trend that increasing concentration of nanoparticles reduced their size. Nanoparticles supplementation has also no pronounced effect on lymphocyte proliferation. At highest concentration, at 80 mg nanoparticles, the lymphocyte proliferation was more as compared to the rest of the groups. The nanoparticles effect on the histology was observed. More pronounced effect was observed on the area of sickle nodules. The area of the sickle nodules was decreased, but their germinal center area was increased. It shows that their improved production produce B and T cells. Finally, we concluded that nanoparticle supplementation increases the germinal central area nodule, thus increasing the efficiency of the immune cell production. But we observed no significant effect on the relative weight of bursite, sickle tonsil, and lymphocyte proliferation assay. From our study, we observed this that iron supplementation is not important to provide in feed. The natural Saturation present in feed is sufficient for growth and as well as for the immunity.
Thank you so much. And for our first poster presentation, we have Banin Abbas from UK, and the title of presentation is Exploring the Therapeutic Potential of Azadirachta indica in Alleviating Ligature-Induced Periodontitis in Rats.
Assalamu alaikum. My name is Dr. Banin Abbas. I have done BDS from Karachi Medical and Dental College in 2020 and currently doing MPhil from Department of Physiology, University of Karachi. Today, I am here to present my research poster on the topic of exploring the therapeutic potential of Azadirachta indica in alleviating ligature-induced periodontitis in rats. This study was aimed to investigate the effectiveness of Azadirachta indica, that is commonly known as neem plant extract, on periodontal infections, highlighting its potential as an inexpensive, risk-free alternative to expensive allopathic medications. As we all know that antibiotic resistance has increased worldwide, so natural herbal remedies provide resistance-free alternative to them. First of all, we all should know that what is periodontitis. Periodontitis is a chronic gum infection that involves tooth and its supporting structures, leading to tooth loss, bone loss, and loss of overall well-being of an individual. It presents with bleeding gums and plaque deposition in initial stages, and in severe cases, tooth mobility due to loss of alveolar bone. It affects around 19% of adult population worldwide, according to WHO Fact Sheet 2022. Herbal remedies like neem are widely used for health benefits that are rich in bioactive compounds found in different parts of a plant. These natural options, rooted in ancient culture and diverse practices, offer prevention and treatment for various illnesses, promoting overall well-being. This study aimed to evaluate the therapeutic potential of Azadirachta indica in alleviating inflammatory cells, that is neutrophils, in the blood of ligature-induced periodontitis rats. As through research, we got to know that neutrophils are the first line of defense in inflammation. So for this, 36 albino vista rats were divided into 6 groups, and each group contained 6 rats. Group 1 remained untreated. In group 2, ligature-induced periodontitis was done by tying 50 silk ligature around mandibular incisors for 7 days. Group 3, ligature-induced periodontitis was done for 14 days. Group 4, Group 5, and Group 6 were treatment groups in which the extract was given. In Group 4, Azadirachta indica plant extract of 300 mg per kg body weight was given orally via gavage after 7 days of ligature placement for 7 days. Similarly, in Group 5, a ligature-induced periodontitis was done for 7 days, and then 500 mg per kg of Azadirachta indica plant extract was given orally via gavage for 7 days. In Group 6, ligature-induced periodontitis was done, and then metronidazole in 100 mg per kg of body weight was given. As we know that metronidazole is the drug of choice for treating periodontal infections. Now, come to this chart. Neutrophils are plotted against y-axis and experimental groups are given on x-axis. This chart shows the effect of Azadirachta indica for 7 days on neutrophils, 2 different doses. For this, when we compare the group 2 with group 1, we can see a rise in neutrophil count, but this rise is non-significant. When we compared group 3 with group 1 and group 2, a rise in neutrophil count can be seen, but this rise is also non-significant. When we compared group 4 with group 1, group 2, and group 3, we can see a reduction in neutrophil count, but this reduction is non-significant. When we compared group 5 with group 1 and group 4, a non-significant reduction can be seen, but a significant reduction can be seen when compared with group 2 and group 3. In group 6, we can see a non-significant reduction in neutrophil count when compared with group 1, group 2, and group 3, but a non-significant increase in neutrophil count when compared with group 4 and group 5. This proves that 500 mg per kg of Azadirachta indica plant extract for 7 days significantly attenuated the infiltration of inflammatory cells, that is neutrophils, in blood of periodontitis-induced rats as compared to other groups. In these pictures, we can see on day 0, when we were tying the ligature, we can appreciate healthy gingiva. On day 7 of ligature placement, we can easily see the bleeding gums. On day 14, we can see bleeding gums plus slight edema around the teeth. And this 500 mg per kg AI Azadirachta indica group, the gingiva was similar to the day 0 gingiva, that is, it was almost normal. So we concluded that Azadirachta indica plant extract has significant effects on reduction of inflammatory cells in blood of periodontitis-induced rats, and that it possesses antimicrobial and anti-inflammatory action. Thank you so much.
Thank you so much, Bunny. And our last poster presenter is Saira Javed from UK, and she will be presenting on antioxidant effect of allium saturn garlic on ligature-induced periodontitis in Western rats.
Assalamualaikum. I am Dr. Saira Javed. I graduated from Karachi Medical and Dental College, and now I am enrolled in MPhil program in Department of Physiology, University of Karachi. I would like to discuss a topic of my research, which is antioxidant effects of Allium sativum, also known as garlic, on ligature-induced periodontitis in Western rats. So what is periodontitis? Periodontitis is the inflammation of gingiva, which is caused by plaque and calculus, which leads to the destruction of periodontal ligaments and bone, resulting in the loss of tooth. It is the most prevalent, 11th most prevalent disease in the world, and as I see in Pakistan, every other individual is suffering from periodontitis. For treatment options, we have scaling, root planning, and antibiotics, but these are treatment options with more side effects and more time consuming and expensive. So we wanted to provide a solution to the public, which is convenient for them and less expensive. And so we designed our research on garlic, as garlic has been well researched for its antioxidant properties and anti-inflammatory properties. So the aim of our study was to investigate the antioxidant effects of garlic, whether it can treat periodontitis or not. For that, we took 36 Wistar rats and divided into 6 groups. The first group was remain untreated. The second group, we tied a ligature around mandibular incisors for 7 days to induce periodontitis. For group 3, we placed the ligature for 14 days. For group 4, we placed the ligature along with the administration of Allium sativum 1 gram per kg body weight for 7 days. For group 5, We provide them with 3 gram per kg body weight of allium sativum for 7 days. For group 6, we tie the ligature and provide them with metronidazole 100 mg per kg body weight for 7 days, as metronidazole is well-researched drug, also known as flagyl, for periodontitis. So we wanted to compare our results, our herb results with metronidazole. After 7 and 14 days, we sacrificed the animals and took their tissue samples and blood for biochemical and histopathological assessment. As we know, during periodontitis, neutrophils levels are raised and inflammatory biomarkers are also raised, which leads to the production of reactive oxygen species and decrease in antioxidant enzymes, which are catalase and superoxide dismutase. This shift in between ROS and antioxidant enzyme cause the ROS burden and ultimately damage. After our results, we see that the catalase levels were reduced in PD7 and PD14 groups, which means during periodontitis, catalase levels are reduced. With our herb, there is a non-significant increase in the catalase level, and with 3 gram of allium sativum, there is a significant increase in catalase level as compared to control. Similarly, we have seen that the SOD levels, superoxide dismutase levels, were significantly decreased in PD7 and PD14 days. And with drug with AS 1 gram, there is a significant increase in SOD levels. And with 3 gram of allium sativum, as you can see here, there is a massive increase in SOD levels. However, with drug with metronidazole, there is no increase in SOD. So we concluded that the treatment with 3 gram per kg body weight of allium sativum for 7 days effectively reduced the ROS burden and increase the antioxidant levels, which means that it can fight off against the periodontitis and provide a less expensive treatment option. Thank you.
Thank you so much, Shaira, and I am really thankful to all of you for joining this session. And I will be giving the closing remarks for this ceremony. So we believe that any work or any activity cannot be completed without a team leader. And for that, we are honored and privileged that we have with us a great leader, Dr. Hasim Anwar and Dr. Ghulam Hussain, and our great faculty team members from our Department of Physiology, UCUF, on all the other faculty members from different other departments and universities who joined us in this conference and helped us in organizing and working everything out. And I also like to thank each of our participants, our presenters, our guest speakers, national, international, our sponsors, collaborators, and most importantly, I am so much thankful and so much proud on the team, the Physiological Society Pakistan. So they worked so hard in organizing this conference, and I'm really super proud. And hopefully, I would like to see on IBBC 2025. We will send you invitation again, and we will be really happy if you could join us again in IBBC 2025. Till then, bye, Allah Hafiz, and we will see you again, inshallah.